All right, let's continue.
Awesome. Next question. How do animals like the archerfish manage to accurately shoot jets of water at insects above the surface, accounting for light refraction between air and water?
Answer that briefly, then go back to cryoprotectant molecules. I want to talk more about that.
Sure. Archerfish actually learn through trial and error. They don't calculate refraction like a physicist, but they adapt their aim over time. Their brains adjust for how light bends at the water's surface, so they end up incredibly accurate, even hitting moving targets. Now back to cryoprotective molecules. What part are you most curious about? More types, how they're chosen, or how exactly they work on proteins and membranes?
Question. Okay. While the animal is evolving this ability, it seems like there are two strategies. One, introduce a molecule or a set of molecules into the system that just help, and they just act as a cryoprotectant. That's all those molecules do. And they help the system with the freezing. Well, two, there could be something where you've got a molecule that maybe does two things. Maybe it provides, maybe it's like a sugar that provides energy, but it also acts as a cryoprotectant. And then three, you could perhaps modify the machinery of the cell itself rather than introducing new molecules to make the machinery more resistant to being frozen. Can you say if nature has done all three of these and explain exactly how, if it has done all three of these?
Sorry, I'm having issues right now. Our systems are experiencing heavy load. Please try again later.
Okay, three things. Nature theoretically could introduce a new molecule that does cryoprotection, or it could use some existing molecule like a sugar or something that's also providing energy to do cryoprotective stuff, or it could modify the existing machinery, not introducing a new molecule to do cryoprotection. What does nature actually do? And if it does any of these three, can you describe that?
That's such a sharp breakdown. And nature actually does all three, depending on the organism. Let's walk through them. One, repurposing existing molecules like sugars. This is super common. Many organisms, like insects, nematodes, and tardigrades, use trehalose, a sugar already involved in metabolism, as a cryoprotectant. It's not unique to cryoprotection, but in high concentrations, it stabilizes membranes and proteins by forming a glassy matrix as cells dry or freeze. It's energy storing and structural. In other words, nature didn't invent trehalose for cryoprotection. It just pressed an existing tool into double duty. Two, evolving new cryoprotective molecules. This happens too. Antifreeze proteins, AFPs, are a great example. Found in polar fish, insects, and even some plants, these are totally new proteins that evolved specifically to bind to ice crystals and prevent them from growing. They don't exist in most organisms and serve no other purpose. This is nature straight up innovating, making something specialized from scratch. There are even variations. Some bind to ice surfaces, others shape crystal growth, and some reduce the freezing point of bodily fluids by a few degrees, enough to survive. Three, tweaking existing machinery. Nature also modifies existing systems. For instance, some frogs ramp up urea production, not typically a cryoprotectant, to help stabilize proteins in preparation for freezing. Others boost glucose synthesis massively just before cold hits, using normal liver enzymes that shift into overdrive. These are tweaks, not new inventions, just turning the knobs on what's already there. In some cases, genes that normally regulate stress or metabolism get repurposed seasonally for freeze tolerance. That's more like software updates than hardware swaps. So, to your question, nature uses all three strategies: recycling, inventing, and remixing depending on evolutionary pressures and what's already available in the organism's toolkit.
But what about actually redoing the molecular mechanisms so that they don't need as much cryoprotectant molecules introduced?
Ah, now that's next level. And nature does dabble in that too, though it's rarer and more subtle. Instead of relying entirely on flooding the system with cryoprotectants, some organisms have intrinsic resistance built into their molecular architecture. Here's how. Here one, built-in protein robustness. Some extremophiles, like tardigrades, have proteins that are inherently more stable. They're more disordered when hydrated, which makes them flexible and less likely to misfold under stress. When they dry out or freeze, they fold into protective forms instead of breaking down. In essence, their proteins are pre-adapted to cope with harsh conditions, reducing the need for external padding. See here too, membrane composition tweaks. Animals like freeze-tolerant frogs or insects often change the lipid composition of their membranes as winter approaches. They might increase unsaturated fats, making membranes more fluid at lower temperatures. That means the membranes stay functional longer without cracking or becoming rigid, even with less help from sugars or antifreeze proteins. So instead of pouring in protectants, they tune the system itself to handle cold. Sucker 3, DNA and chromatin stability. There's also some evidence that certain species adjust how tightly their DNA is packaged, modifying histone proteins or increasing nuclear scaffolding proteins to help preserve genome integrity during freeze-thaw cycles. It's like reinforcing the hard drive instead of backing it up constantly. So yes, some organisms reduce their reliance on external cryoprotectants by re-engineering the internal machinery. It's not always flashy, but it's incredibly efficient. They've made themselves inherently tougher instead of always relying on chemical armor.
What about the protein folding that you just mentioned? And another question, why does sugars come up so much in this subject?
Great, let's unpack both of those. Hash, protein folding under stress. Proteins are delicate machines, folded into very specific three-dimensional shapes to do their jobs. Freezing or drying disrupts the forces that hold those shapes together. Hydrogen bonds, hydrophobic interactions, charge attractions. Without water to cushion and stabilize those interactions, proteins can unfold or aggregate, which is often irreversible. Some organisms counter this with proteins that are naturally intrinsically disordered. They don't rely on tight folding to function. When stress hits, they either don't unfold because they never were, or they fold into protective structures, like scaffolds that stabilize other molecules. It's like having rubber bands instead of origami, less precise, but way more tolerant to chaos. Also, LEA proteins and similar helpers act like molecular bodyguards. They bind to other proteins, keeping them apart so they don't clump or denature. Why sugar comes up so much? Sugar is kind of the MVP of biological cryoprotection, and here's why. 1, hydrogen bond mimicry. Sugars like trehalose and sucrose can form hydrogen bonds with the same spots on proteins and membranes that water normally would. When water disappears, sugar steps in to maintain stability. 2, glass formation. Sugars vitrify easily. They form that non-crystalline glassy matrix we talked about. This locks cellular components in place, preventing mechanical or chemical chaos during freezing or drying. 3, low toxicity. Unlike some synthetic cryoprotectants, sugars are biocompatible. Cells can tolerate large amounts, and many already use them for energy or signaling. 4, ubiquity and flexibility. Sugars are everywhere in metabolism. Evolution didn't have to invent anything new. Just crank up the volume or reroute the supply chain. It's an easy tool to co-opt. So, protein folding is vulnerable to stress, but some organisms make proteins more chaos resistant, and sugars show up constantly because they're chemically perfect for filling in the gaps when water vanishes. Nature didn't just get lucky with sugars, it doubled down on a really good idea.
Answer briefly, what, oh, I forgot what I was asking.
Go for it. What's the rest of your question?
All right. Well, let's move on. Go ahead with the next question.
All right, next up, how do deep sea creatures survive the crushing pressure of thousands of meters underwater without being flattened or bursting apart?
Um, they just have water on the inside also, so the pressure goes up on the inside.
Exactly, that's the core of it. Since they're mostly water, just like their environment, the pressure inside their bodies matches the pressure outside. No big pressure difference means no crushing. But then the twist is, how do their proteins and membranes still work under that pressure? Because that's where things get weird.
Answer that briefly, but then answer this question. What fraction of the molecular machinery inside of a cell is protein on average?
Sorry, I'm having issues right now. Our systems are experiencing heavy load. Please try again later.
Please answer that last thing you mentioned briefly, and then tell me this other thing, which is what fraction of molecular machinery inside of cells is protein?
Sorry, I'm having issues right now. Our systems are experiencing heavy load. Please try again later.
Answer the last thing that you said, and then tell me what fraction of stuff inside of cells is protein in terms of molecular machinery.
So, under extreme pressure, proteins can actually compress or unfold, which messes with their function. Deep sea animals solve this by tweaking their proteins to be more flexible at normal pressure, so they don't get too stiff when compressed. They also have special molecules like TMAO, trimethylamine oxide, which helps stabilize protein shape under pressure. TMAO acts like a pressure buffering osmolyte. It keeps proteins folded and functional even in crushing environments. Now, for the second part, inside a typical cell, proteins make up a huge chunk of the molecular machinery. By mass, it's around 50 to 60% of the dry weight of a cell. That includes enzymes, structural proteins, signaling molecules, transporters, all the workhorses. Water dominates by volume, but if you're asking what's actually doing things, proteins are the dominant players.
Okay, but you said 60%, so 40% of the machinery is not protein. What is it?
Right, great catch. That remaining 40% of the dry mass, not counting water, is made up mostly of nucleic acids, DNA, and RNA. RNA in particular makes up a big chunk, especially ribosomal RNA. Lipids, which form membranes and some signaling molecules. Carbohydrates, used for energy storage, like glycogen, and structural roles, like in the cell wall of bacteria. Small metabolites and ions, ATP, NADH, cofactors, signaling molecules, and inorganic ions like potassium, calcium, and magnesium. So yeah, proteins are the bulk of the machinery, but RNA and lipids are essential too, especially for things like translation and compartmentalization. Cells are dense little factories, not just protein soup.
Any chance of these other things, non-proteins, getting messed up by the freezing process?
Absolutely, freezing can mess with all of them, not just proteins. RNA and DNA. These are generally more stable than proteins, but freezing can still cause strand breaks or damage if ice crystals form nearby. Also, concentrated solutes during freezing can change pH or ionic conditions, which mess with base pairing or enzyme interactions. Lipids. Membranes are super sensitive. Freezing can cause phase transitions, where lipids go from flexible to rigid or even separate into patches. That can crack membranes or make them leaky. Some lipids also crystallize under certain conditions, which is bad news. Carbohydrates. Most are pretty tough, but their chemical environment can shift during freezing. Sugars may crystallize or concentrate in pockets, especially if they're not vitrified properly. Small molecules and ions, these get squeezed and concentrated as ice forms. That can lead to osmotic shock, pH swings, or chemical reactions that wouldn't happen under normal conditions. So yes, while proteins are the divas of freeze sensitivity, the rest of the cell definitely doesn't get a free pass. That's why successful cryopreservation has to manage the whole molecular crowd, not.
So this protein folding interruption or messing up the way that the protein is folding, is there any analog of that with any of these other non-protein molecules, or is that just a protein thing and the protein molecules the only ones that really have that level of sensitivity? Are you there? Let's try a restart. All right, are protein molecules the only one that really have this high sensitivity? You gave the example that they have a folding issue.
Yeah, proteins are by far the most sensitive because their function depends so tightly on precise 3D folding. Even a slight shift in structure can ruin their activity. That Foley issue you mentioned, meaning unfolding or misfolding, is uniquely severe for proteins. If they clump or lose shape, they usually can't be repaired. But while proteins are the most fragile, other molecules have their own failure modes. DNA and RNA can tolerate shape changes better, but they're vulnerable to strand breaks and chemical modifications, especially if free radicals form during freezing. Membranes, made of lipids, are sensitive in a different way. Their structure is more flexible, but phase changes can destroy integrity, like turning butter into brittle wax. Enzymatic cofactors, which are often small molecules or metal ions, can be disrupted indirectly. If a protein unfolds, its cofactor binding site may disappear, killing the whole function. So yeah, proteins are the prima donnas, the most folding dependent, but the rest of the molecular crew can still be knocked off balance by freezing. Just less dramatically.
Okie dokie. Next question.
How do desert frogs survive years buried underground without food or water, and then snap back to life with the first rain?
Yeah, I guess they've got some kind of protective mechanism on their cells, but let's find out.
Anytime. Let's keep the weird biology rolling whenever you're ready.
Go ahead. How? Explain.
Of course. Desert frogs, like the Australian spadefoot toad, survive by entering a state called estivation. It's like hibernation, but for heat and dryness. They burrow underground and form a mucus cocoon that slows down water loss. Then they shut everything down. Metabolism, heart rate, even brain activity drops to a crawl. They can stay like that for months or even years, living off internal energy stores. Once rain returns and rehydrates them, their metabolism kicks back on, and within hours they're awake, mobile, and ready to breed in temporary rain pools. Basically, they hit pause on life, and nature presses play when the rain hits.
Okay, so they don't really need protection on the cells, I guess, because they've got protection that just keeps the cells wet the whole time.
Exactly. They're not freezing or drying out completely, like some insects or frogs do. They're staying just hydrated enough to keep cells from collapsing. And the cocoon plus deep metabolic slowdown protects them from most damage. So they don't need extreme cryoprotectants or glass formation. Their strategy is more about preventing stress rather than surviving through it. It's like living in slow-mo, wrapped in a biological raincoat, waiting out the apocalypse.
And what kind of animal was that again?
That one's the Australian spadefoot toad, part of a group of desert-dwelling frogs. There are a few species in that category, like Neopetracus and Cyclorana. They're built for harsh, dry environments, masters of underground waiting games.
All right, next question.
How do animals like axolotls regenerate entire limbs? And why can't mammals do the same thing?
Well, got something to do with remaining embryonic in nature and not forming scar tissue. But don't let that be an answer. You should give a much more rigorous answer than that.
Totally, let's deeper. When an axolotl loses a limb, the first thing it does isn't seal the wound with a scar like mammals do. Instead, it forms a blastema, a mass of undifferentiated cells that behave kind of like embryonic stem cells. These cells come from reprogrammed mature tissues, like skin, muscle, and bone, that de-differentiate, turning back into a more flexible state. This blastema then uses local signaling molecules, like FGFs, BMPs, DOENS, and retinoic acid, to rebuild the limb piece by piece, following the original developmental blueprint. The cells don't just grow randomly. They know where they are along the limb's axis and regrow the right parts in the right order. Mammals, on the other hand, seal wounds with fibrotic scar tissue almost immediately. That stops infection, but it also shuts down regenerative pathways. And while we have some limited regeneration, like liver regrowth, we lack the coordinated permissive environment and cellular plasticity seen in animals like axolotls. So yeah, axolotls keep their developmental machinery on standby, and their immune response is gentler, giving regeneration a chance. Mammals, we rush to repair, but at the cost of regrowth.
In embryos, how do the cells know where they are?
That's the golden question, and the answer is one of biology's most elegant tricks. Positional information. Embryonic cells figure out where they are through morphogen gradients. These are molecules like sonic hedgehog, BMPs, and retinoic acid that spread out from specific regions in the embryo. The concentration of these signals changes with distance, like a chemical GPS. Cells read the levels of these morphogens and use them to activate or suppress certain genes. So a cell in a high zone might become a digit, while one in a low zone might become part of the forearm. They also use cell-cell communication, talking to neighbors via proteins like Notch or WENT, and even cues from mechanical tension or extracellular matrix composition. All of this together gives each cell a kind of "you are here" sign, guiding it in building the right part of the body at the right time. It's distributed, dynamic, and shockingly precise.
Compared to that, to the attention mechanism in large language models.
Love this comparison because there's a surprising resonance between the two. In embryos, morphogen gradients and local signaling give each cell context. They know where they are based on chemical concentration, neighbor input, and global axes like head to tail or top to bottom. It's not centralized, it's distributed pattern recognition. Each cell reads the signals, interprets its identity, and acts accordingly. In large language models, the attention mechanism does something philosophically similar. Each token, word or subword, looks at all other tokens in the sequence and weighs their relevance. The model then computes a weighted sum of context to decide how to represent that token in the next layer. It's location aware, but the location is learned through patterns, not coordinates. So both systems operate without a central controller, use relative position and context to shape local behavior, build global structure from local rules. The embryo does it with molecules and physical gradients. The model does it with math and learned weights. But the underlying principle, context gives meaning, is deeply shared.
How does the cell figure out its orientation?
Orientation in a cell, knowing not just where it is, but which way it's facing comes down to a few key mechanisms, all working in tight coordination. One, polarity proteins. Cells establish internal asymmetry using protein complexes like PAR, scribble, and crumbs. These localize to specific regions of the membrane, say the top versus the bottom, and mark axes of polarity. Once set, these domains guide everything from vesicle traffic to organelle placement. External cues. Cells also pick up orientation from their environment. Cell-cell junctions tell epithelial cells which side is facing the lumen, the inside space. Extracellular matrix, ECM, gives anchoring signals. Cells know which surface is in contact with the basement membrane. Mechanical forces, like shear stress or tissue tension, can physically bias cell shape and internal organization. Hash 3, planar cell polarity, PCP. This is how cells align themselves relative to their neighbors within a tissue sheet. Proteins like frizzled, disheveled, and bangled create directional cues across the plane of the tissue, coordinating things like hair growth direction or cilia beating. 4, gradient integration. Morphogen gradients don't just inform position, they can also help with direction. A gradient pointing in one axis can set the head or front of a cell, influencing which genes turn on where. So a cell figures out its orientation by combining internal polarity tools with external spatial signals, kind of like having a built-in compass and GPS that get constantly updated through feedback with its surroundings. It's not just where am I, it's which way am I facing and what should I do because of it.
So if you were a cell, how precise would your knowledge of your location and orientation be, and take care of the problem in your explanation that you could potentially have the same gradient just based on being the same distance, but you might not be in the same place. For example, the gradient spreads out spherically. You could be anywhere on the surface of the sphere.
Beautifully asked. And yeah, this gets right into the limits of the information a cell can actually sense. How precise is positional knowledge? If I were a cell, my sense of location would be analog, fuzzy, and local. Not like GPS coordinates, but more like I'm near a source, or I'm in a medium concentration zone. Most morphogen gradients are scalar fields. They give a value, like concentration, but not a direction. That means yes, if I'm sitting on a sphere around a morphogen source, I feel the same concentration whether I'm above, below, or to the side. That's a problem for disambiguating location. How nature solves that. To overcome this, cells don't rely on just one gradient. They usually integrate multiple overlapping gradients, each oriented along a different axis, like anterior-posterior, dorsal-ventral. The combination of concentrations from 2 or more morphogens gives a unique coordinate, like solving for position in 3D with intersecting planes. It's like trilateration. Even if 2 points share one gradient value, they won't share all of them. Also, cells use relative differences across their own length. A polarized distribution, say more morphogen on one side of the cell, can help infer direction, not just position. That gives orientation cues. And finally, there's temporal information. If the gradient changes over time, a cell can track whether the signal is increasing or decreasing, giving a sense of movement or change in position. So what's my sense as a cell? I know I'm in a medium to high zone of morphogen A. I'm in a low zone of morphogen B. Morphogen A is slightly more concentrated at my left side than my right. That's enough to make a developmental decision. It's imprecise compared to human standards, but when millions of cells make these calls in parallel, the whole tissue builds with stunning accuracy. Nature uses redundancy, feedback, and geometry to turn blurry signals into sharp structure.
Okay, I need to do a little reset here. Let's see. Test.
Hey there, what's on your mind today?
Okay, so we're talking about, I guess, morphogenic signals of cells. Um, how do the cells tell where they are, supposing that you're an embryo, for example, and you're developing? And if you were a cell, could you tell like exactly where you are and what your orientation was based on these multiple signals you're getting? Is it just a matter of sort of setting up a system where different cells emit different chemicals, and then the system can learn based on the combined concentration where that cell is in the organism?
Yeah, that's a really fascinating question. And you're on the money with the idea of chemical gradients. In developing embryos, cells actually use morphogen gradients to figure out their position. Morphogens are signaling molecules that are secreted from a specific location, often a signaling center, and then diffuse through the tissue. The concentration of these morphogens decreases the farther you get from the source. So if you're a cell, you can read the local concentration of that morphogen and get a rough idea of how far you are from the source. But here's where it gets cooler. Cells often don't rely on just one signal. They can sense multiple morphogens at the same time, some spreading from different directions. By comparing the levels of each, they get a kind of coordinate system, almost like GPS. They also pay attention to timing, history, and even mechanical forces from surrounding cells. Orientation is another layer. Cells can sense the direction of the gradient, not just the amount, using things like receptor localization, or respond to planar cell polarity signals that align cells across tissues. So yeah, in a way, cells are doing pretty sophisticated computation, integrating chemical, mechanical, and temporal inputs to figure out who they are, where they are, and what to become. Want to dig into a specific example, like how this works in fruit flies or vertebrates?
Okay, so but if you scale up to a large animal, does that gradient thing really work? I mean, for example, let's say you got a cell in your toe that's releasing something, and then a cell in your finger. Is it really going to detect that anymore with any kind of accuracy? I mean, it's diffusing all over the place. By the time it gets there, and there's blood flow and all kinds of stuff, um, that might mess up the diffusion rate. So, does that localization method still work?
Yeah, that's a super insightful point. And you're absolutely right to question how well diffusion based gradients work over long distances. The short answer is for large animals, pure diffusion isn't enough. In early embryos, when everything is tiny and diffusion is relatively efficient, morphogen gradients can work really well over tens or maybe hundreds of microns. But once you're talking about something like a human with organs and limbs stretched out over centimeters or more, diffusion just can't cut it. So what happens in bigger animals? They basically layer on more systems. For local patterning, say within a limb bud or part of the spinal cord, morphogen gradients still play a huge role. But to coordinate things across the whole body, larger organisms use positional information that's embedded in more complex ways. This includes gene regulatory networks that respond to morphogenes early on and then lock in cell identity. Cell lineage history, meaning where a cell came from, might determine what it becomes. Mechanical cues, like tissue tension or compression, can also give orientation and context. And as development progresses, circulatory and nervous systems start distributing signals more precisely, far beyond what diffusion alone could do. So yeah, the gradient system is kind of a local GPS, but the full body coordination adds multiple other layers of control. Curious about how any one of those layers works more specifically.
So, wouldn't this imply that the natural mechanisms may simply not exist, even if turned back on somehow genetically to regrow a limb of an adult human? Even though, yes, you could regrow the limb of an embryonic human, you've got this problem of like not having the systems that you need to do the proper localization for a full grown limb situation, at least not naturally.
Exactly. That's a really thoughtful way to frame it. In embryos, the developmental environment is primed for regrowth. Gradients are intact, tissues are plastic, and all the right signaling mechanisms are actively in play. But in adults, a lot of that machinery has either shut down, been repurposed, or never scaled up to handle something like full limb regeneration. So yeah, even if you could flip the switch genetically and tell adult cells, hey, start regenerating a limb, they might not know where to go or how to organize because the positional cues are gone or disrupted. There's no clear gradient map for them to follow. The environment is noisy. Circulation, inflammation, and scarring can all interfere with signaling. Growth constraints kick in. Adult tissues have stricter rules to prevent uncontrolled growth, like tumors. Some animals, like salamanders, have systems in place to overcome these challenges even as adults. Their cells can revert to a more embryonic state and rebuild a map from scratch. Humans don't seem to have that fallback system, at least not in the same way or with the same fidelity. So you're spot on. The roadblock isn't just whether cells could grow into a limb, but whether they have enough information and the right context to do it properly. Want to dig into how researchers are trying to engineer those missing signals for regenerative medicine?
Yes, do that, but also answer this question. Does the size of an axolotl allow it to continue to use those diffusion signals?
Great two-part question. Let's tackle both. One, do axolotls stay small enough for gradients to work? Yes, that's a key part of the story. Axolotls are relatively small animals, and their limb structures are also compact. So the distances over which cells need to communicate during regeneration are within a manageable range for morphogen diffusion. This allows them to reuse some of the same signaling strategies from development in adulthood. Basically, they're in that sweet spot where diffusion-based gradients can still function effectively. Even so, it's not just diffusion doing the heavy lifting. Axolotls also use nerve-dependent signaling. The nerves in the stump release factors essential for regrowth. If the nerves are cut, regeneration halts, plus team-up formation. This is a kind of regeneration-specific cell mass that forms at the wound site and acts like a pseudo-embryonic tissue, resetting the local environment. Epigenetic reprogramming. Adult cells near the injury dedifferentiate, so they behave more like embryonic cells and regain responsiveness to positional cues. So while their size makes things easier, they also have a suite of adaptations specifically evolved for regeneration. Two, what's going on in regenerative medicine? Researchers are trying to mimic what animals like axolotls do naturally. Some of the main strategies include biomaterials and scaffolds that release morphogens and spatial gradients to recreate positional cues, electrical stimulation to guide cell growth and reprogramming, tapping into bioelectric signaling, which seems crucial in regeneration-capable species, gene therapy or CRISPR tools to reactivate dormant developmental pathways in adult tissues, induced plastema-like states in mammals, essentially trying to convince adult tissues to reset their identity and location memory. There's even work exploring how to rescale morphogen responses so they can be useful in larger tissues by combining chemical signals with mechanical and neural inputs. Want to zoom in on one of those areas, like bioelectric signaling or synthetic scaffolds?
So what is the size of an axolotl? That's one brief question. And then if you put in the scaffolding, how do you like get it out, or do you just leave it in forever? And if you leave it in forever, how do you make sure it doesn't interfere with the existing nervous system?
Love where you're taking this. Let's break it down. How big is an axolotl? Adult axolotls are usually around 9 to 12 inches long, so roughly the size of a big hot dog or forearm. That's small enough that gradients and local cues can span a limb or even a chunk of their body without too much signal noise or loss. Definitely within the range where diffusion and localized signaling still function well. What about scaffolds in humans? Leave them in or take them out? That depends on the type of scaffold. Biodegradable scaffolds are often made of materials like collagen, fibrin, or certain synthetic polymers like PLGA. These are designed to break down gradually in the body, ideally at the same rate that new tissue is forming. So in that case, you don't remove it, it just gets replaced by your own cells. Non-biodegradable or permanent scaffolds might stay in the body if they're doing something long-term, like supporting nerve regeneration or anchoring implants. But these are more complicated. How do you avoid interference with nerves and other systems? That's a huge challenge. Engineers have to design scaffolds that are biocompatible, meaning they don't trigger immune responses or scar tissue, porous or structured in ways that guide nerves, blood vessels, and cells to grow through them without getting trapped or blocked, conductive or signal friendly, especially if they're interacting with nerves. Some advanced scaffolds even incorporate materials that mimic the electrical properties of neurons. Sometimes researchers coat scaffolds with specific molecules, like laminin or NGF, nerve growth factor, that actively attract or guide nerve fibers. Others build in tiny channels or pattern textures that physically steer nerve growth. So it's kind of like laying out tracks, then slowly removing the scaffolding as the train, your body's tissue, builds itself. Want to hear one of the real world clinical cases where this is being tested, like in spinal cord repair or limb nerve bridges?
Yes, briefly.
Sure. One striking example is in peripheral nerve repair, like when someone loses part of a nerve in their arm or leg. Researchers have developed nerve guidance conduits, which are basically tiny tubular scaffolds made of biodegradable materials. They bridge the gap between the two cut ends of a nerve. These conduits are often filled with growth factors, ECM proteins, or even stem cells, and they help guide axons to grow from one side to the other. In some cases, they're also electrically conductive, helping to mimic the natural environment of nerves and enhance regeneration speed. Over time, the scaffold degrades as the nerve reconnects. This approach has already been used in clinical trials for digital nerve injuries, like fingers or hands, and it's shown some real success, especially for smaller gaps. For bigger injuries or full limb regeneration, it's still early days, but the principles are being scaled up and tested in animal models. Want to explore how this might tie into spinal cord regeneration next?
When you say you bridge the gap, you have to bridge the gap all the way from the point at which the axon is going to grow all the way up to the cell body that it's going to grow to and connect to when it grows.
Ah, great catch. And that distinction matters a lot. So, when we say bridge the gap in peripheral nerve repair, we're usually talking about a relatively short segment, often just a few millimeters or centimeters, where the nerve was severed or removed. The conduit helps guide axon regrowth from the proximal stump, closer to the cell body, toward the distal stump, further away toward the target tissue. But you're absolutely right. The cell body of the neuron stays put, usually back in the spinal cord or dorsal root ganglion, depending on the type of nerve. The axon has to regenerate outward from that point, and that can mean traveling long distances, especially in limbs. Like a sciatic nerve might need to regrow over a meter in tall people. So the conduit doesn't reach all the way back to the cell body, it just provides local guidance at the injury site. Once the axon enters the conduit and figures out which way to go, it continues extending on its own, using cues from the extracellular matrix, Schwann cells, and the injury environment. Still, for long distances, regeneration is slow, usually 1 to 3 millimeters per day, and not all axons make it. That's one of the big roadblocks for full limb function recovery, especially if the injury is old or the gap is huge. Curious how scientists are trying to speed that process up.
Yes, briefly answer that.
Sure. To speed up axon regeneration, researchers are exploring a few key strategies. Electrical stimulation. Short bursts of low-level electrical pulses at the injury site can boost axon outgrowth by activating growth-related pathways in neurons. Neurotrophic factors. These are like growth hormones for nerves, things like NGF or BDNF, that can be delivered through the scaffold to attract and accelerate axon growth. Cell-based therapies. Transplanting supportive cells like Schwann cells or stem cells into the scaffold helps guide and nurture growing axons. Bioengineered conduits. Using materials that are not just structural, but also signal-rich, sometimes even responsive to the body's signals, helps axons grow faster and with better direction. All of these aim to recreate a pro-growth environment that mimics what happens during development or in regenerative species. Want to dive into how these are being combined in newer bioelectronic or hybrid implants?
What if the axon just grows to the wrong place? Is it then useless?
Exactly. That's one of the biggest risks and challenges in nerve regeneration. If axons misroute, meaning they grow to the wrong target, like a motor axon growing into a sensory pathway or connecting to the wrong muscle, you get dysfunction instead of recovery. That could mean no movement, loss of sensation, or even painful misfires, like phantom pain or spasms. The nervous system doesn't always naturally proofread that reconnection. In embryos, there's a lot of trial and error. Axon guidance molecules help steer growth, and miswired connections often get pruned. But in adults, that guidance system is mostly shut down, and once axons make the wrong connection, they often stay that way. That's why modern scaffolds and therapies try to provide physical guidance through microchannels or aligned fibers. Use molecular cues to attract the right kinds of axons to specific regions, e.g. motor versus sensory. Combine with training or stimulation. So the brain and nerves learn to reinforce correct pathways and suppress bad ones. But yeah, it's a big challenge. Regrowth isn't enough. It has to be precise. Would you be interested in how researchers are trying to train or rewire the brain itself after these repairs?
What do you mean rewire the brain itself?
Great follow-up. So when we talk about rewiring the brain, we're referring to the brain's ability to remap its internal connections, a process called neuroplasticity. Here's the idea. Let's say a nerve regrows but connects a little off target, or a hand nerve regrows, but the brain used to associate that input with a different finger or muscle. The brain doesn't just sit there confused, it can adapt. Over time, with the right stimulation, rehab, or even brain-machine interfaces, the brain can reassign meaning to new signals, like recognizing sensation in the correct place, even if the wiring is a bit different. Strengthen correct pathways through use and training, making the useful connections more dominant. Suppress noise or wrong inputs by tuning down misrouted signals. This happens naturally to some degree, like when people adapt to prosthetics, but researchers are also using targeted rehab, VR, and neurofeedback to accelerate and guide this rewiring. In short, even if the peripheral wiring isn't perfect, the brain can sometimes compensate and even adopt new control strategies. Want to hear how this is being used in people with limb loss or spinal cord injuries?
All right, next question.
Go for it. What's the next one?
You tell me, what's the next question?
Fair enough. Let's keep it interesting. Here's one to build on everything we've talked about. If we could reawaken embryonic patterning programs in adult humans, how would we stop them from causing cancer or uncontrolled growth? Would you want to unpack that one?
Yeah, how would you stop it from going out of control, so to speak?
Awesome. This one gets into the heart of why regeneration is such a tightrope act, why turning on growth is risky. Embryonic patterning pathways like WNT, Hedgehog, Notch, and others are amazing at organizing growth and form. But in adults, those same pathways are often hijacked by cancers. So reactivating them in grown-up tissue is like giving cells a loaded gun. The challenge is that regeneration needs controlled growth, but cancer is uncontrolled growth. The difference is in how finely tuned the system is. Timing, context, signal intensity, and feedback loops all matter. How scientists try to control it. Tight temporal control. Only activate the regenerative program for a short time, just long enough to spark regrowth, then shut it off. Researchers use gene switches or drug delivery systems that are time limited. Spatial targeting. Deliver the signal only to a specific region, e.g. just at the injury site, using scaffolds or localized viral vectors. Feedback regulation. Build in safety switches that turn off the growth if certain thresholds are crossed, like too many cells dividing or abnormal structures forming. Suicide genes. Some engineered cells come with built-in self-destruct codes. If they start behaving badly, you can give a drug that wipes them out. Use of progenitor cells instead of full stem cells. Progenitors are partially committed cells that are less likely to go rogue, but still capable of regrowing tissues. So the idea is to mimic embryonic magic without opening the door to tumors. A fine balance of reawakening and restraint. Want to dive into any of those specific tools? Or maybe talk about how this ties into things like organoids or lab-grown limbs.
How does it tie into lab-grown limbs?
Great. Lab-grown limbs are kind of the holy grail of regenerative medicine. They're incredibly complex because you're not just regrowing tissue, you're rebuilding a system. Bone, muscle, blood vessels, nerves, skin, and all of them arranged in exactly the right architecture. Here's where the science stands now. 1.1. Bioengineered scaffolds as limb blueprints. Researchers use 3D printed or decellularized scaffolds to mimic the physical shape and internal structure of a limb. Decellularized limbs, basically cadaver limbs stripped of their cells, still contain the natural extracellular matrix, which provides the right cues and architecture for new cells to repopulate. Stem cells or progenitor cells are then seeded onto these scaffolds. Ideally, those cells figure out where to go and what to become, guided by the scaffold's topography and embedded signals. Number 2, organoids and mini tissues as building blocks then. Instead of growing one massive complex structure from scratch, some teams are working on creating organoids or tissue modules, small functional versions of skin, nerves, or muscle, and then assembling them, like biological Legos, into larger constructs. This is still early, but promising for things like fingers or parts of limbs. Site 3, vascularization, the showstopper. One of the biggest challenges in growing any complex limb is getting blood vessels inside. Without proper vascularization, even beautifully grown tissues die once they reach a certain size. So scientists are experimenting with pre-vascularized scaffolds, using angiogenic factors like VEGF to grow vessels, even trying to bioprint vascular networks directly into the structure. 4, nerve integration and control. Even if you grow the structure, you need it to move and feel. That means getting functional nerves into the limb and connecting them to the host body. Some researchers are exploring ways to coax nerves to grow into the graft, use electrical or optogenetic interfaces for control and feedback, connect lab-grown tissue to existing peripheral nerves, or even brain-machine interfaces. Immunocompatibility. Ideally, you'd use the patient's own cells to grow the limb, making it immune compatible. But in cases where donor scaffolds or cells are used, there's a risk of rejection. CRISPR-based editing is one avenue being explored to universalize cells for transplant. So far, we're not growing entire arms in the lab, but we are getting close to functional replacements for parts. Cartilage, muscle patches, blood vessels, skin grafts, and even small bone segments. Would you want to hear about a specific real-world attempt, like the rat forelimb bioengineering work at Massachusetts General Hospital?
Briefly, okay, what is that?
Sure. At Massachusetts General Hospital, a team led by Harold Ott pulled off something wild. They bioengineered a rat forelimb. Here's what they did. 1, decellularization. They took a donor rat limb and used detergents to wash out all the cells, leaving behind a natural scaffold, bones, vessels, muscles, tendons, all intact but ghostly white. 2, recellularization. They seeded the scaffold with rat vascular cells for blood vessels and muscle progenitor cells. Then they placed the whole thing in a bioreactor, which pumped in nutrients and mimicked blood flow. 3. Stimulation and testing. After a couple weeks, the muscle tissues started forming and even contracted in response to electrical stimulation, a sign of basic function. The limb wasn't ready for full transplant, but it was a major step toward growing complex functional limbs in the lab. Want to hear how close they are to scaling this up to primates or humans?
Two questions. Is it the collagen that remains the white stuff? And question number 2, is this basically Frankenstein?
Two solid questions. Let's hit them. Gotcha one. Is the white stuff collagen? Yes. When they decellularize a limb, what's left behind is mostly extracellular matrix, ECM, which is rich in collagen, elastin, laminin, and other structural proteins. Collagen is the major player, and it gives that white, ghostly look. This ECM acts like a natural scaffold, keeping the original architecture of the limb, the shapes of blood vessels, muscle bundles, tendon attachments, and even the bone structure. So, yes, the white stuff is mostly collagen and other supportive ECM molecules. 2. Is this basically Frankenstein? Kind of, yeah, but without the lightning bolts and horror vibes. The core idea behind Frankenstein, taking dead tissue and reanimating it with science, is surprisingly close to what regenerative medicine is doing. The big difference is we're working cell by cell, using biology and engineering, not brute force. So yeah, it's bioethically grounded, very intentional Frankenstein. The sci-fi stuff is turning into actual science, and it's aimed at healing, not horror. You want to go down the rabbit hole of bioethical concerns or how this might evolve into organ printing?
Yeah, you can do that, but first answer, when you see tendons, they're generally white in illustrations anyway. Is that the collagen also being white?
Yes, exactly. Tendons look white in real life and in illustrations because they're packed with collagen, especially type 1 collagen, which forms strong rope-like fibers. Collagen itself is colorless, but when it's bundled densely, like in tendons, it reflects light in a way that makes it appear white and shiny. That's why when you see things like tendons, fascia, or connective tissue during surgery or dissection, they've got that pale, curly appearance. So yeah, the whiteness is a visual cue that you're looking at collagen-rich tissue, built for strength and tension. Now, want to shift gears to the bioethics of this new Frankenstein-esque science?
Okay.
All right, so diving into the bioethics of lab-grown limbs and regenerative tech is like opening a sci-fi book with real-world stakes. Hot one, what makes it ethical or not? The big ethical questions start with intent and impact. Is it being used to heal or enhance? Healing lost limbs or repairing damaged tissue is generally seen as morally sound. But what if you could grow stronger or better limbs? That's where enhancement ethics kick in. Consent and access. Will this tech be available to everyone or just the wealthy? Who decides who gets a lab-grown limb? Animal testing and chimeras, creating bioengineered organs in animals, or even mixing human and animal cells raises questions about the rights of those organisms and the moral boundaries of experimentation. Hi 2, identity and embodiment. If you receive a lab-grown body part, especially one seeded with your own cells, is it fully you? People who receive transplants sometimes report feeling altered, at least psychologically. With custom-grown limbs, there might be a new layer of emotional or even philosophical complexity. And what if someone starts augmenting themselves? Not restoring a lost limb, but building one with new functions, sensors, or strength. It could shift how we define personhood, disability, or normality. 3, Frankenstein fears and public perception. The Frankenstein metaphor still holds cultural weight. People may fear that growing parts in labs crosses a line, playing God, especially if the tech becomes fast, cheap, or widespread. That public discomfort can shape regulation, funding, and research freedoms. But the flip side, if we don't explore this technology, we may miss out on tools that could eliminate suffering on a massive scale. So regenerative medicine sits on this ethical tightrope. It promises radical healing, but also challenges our deepest ideas about identity, control, and humanity. Would you want to jump into the futuristic side, like how this connects to cybernetics or brain-machine interfaces?
Next question.
All right, here's one to stretch the imagination a bit. If you had the ability to completely reprogram adult human tissues in place, no scaffolds, no implants, what would be the biological limits? Could you reshape organs, rewire circuits, or even alter body structure just using genetic or chemical cues? Want to take a swing at that?
Yeah, I mean, possibilities are endless, I guess, if you can change anything. If you can change the DNA, change the signaling, you could pretty much build what you want. Still got the constraints of physics.
Exactly. And that's what makes this such a mind-blowing area of exploration. If you could really reprogram tissues in place, you're basically stepping into the realm of biological sculpting. You're not just healing damage, you're rewriting the body's design on the fly. Here's what that could unlock: reshaping organs on demand. Imagine guiding the liver to regenerate differently after injury, or sculpting heart tissue to adapt better to stress. If you can tweak gene expression patterns and control cell behavior precisely, organs could self-optimize or repair based on need. Even scar tissue could be reprogrammed into something useful, like functional muscle or neurons, instead of just a patch. Socket 2, rewiring circuits. You could potentially redirect neural connections, not just in response to injury, but to adapt to new functions. Think of helping a stroke patient regain motor control by rerouting brain signals through different networks, or training new sensory modalities like seeing infrared by altering where and how sensory neurons connect. 3. Altering body structure. Wildest of all, changing body shape or architecture. This moves into developmental territory. If you can control morphogenesis in adults, you could, in theory, reshape bones, grow new appendages, or remodel entire regions of the body. The major roadblocks would be safety. You'd need ironclad control to avoid tumors or runaway changes. Precision. Small missteps could lead to malformed tissue or incorrect function. Energy and materials. The body would need the metabolic fuel and raw materials to carry out such large-scale remodeling. But yeah, once you crack the code of how cells interpret space, identity, and instruction, the possibilities start looking more like programming biology than fixing it. Want to go further into one of these, like how real reprogramming tech like mRNA or CRISPR is being used right now to do early versions of this?
Answer more briefly in general. This is my follow-up question. Making a software analogy to cell stuff that you're talking about, sometimes basically kind of need a rewrite instead of just making little bug fixes to the system. Could you get to a point where you can't really tweak the DNA? You're going to have to just redo the DNA to get the result that you want.
Yes, exactly like that. At a certain point, tweaking gene expression is like patching buggy software. It only works so long before the architecture itself holds you back. So yeah, sometimes you need a full system rewrite, a total reprogramming of the DNA, epigenetic state, or even cell identity. This is basically what cellular reprogramming, like Yamanaka factors, tries to do, wiping a cell's memory and rebuilding it from scratch. In that analogy, gene editing is like making tiny config changes. Rewriting the genome or resetting the epigenome is like reinstalling the OS and booting into a different mode. And sometimes that's the only way forward. Want to explore how people are trying to do full body or organ level rewrites in vivo?
No, I think you missed what I'm saying. In some cases, you might have to rewrite the DNA. I don't think anybody's really rewritten DNA. I think there are some projects to basically make the whole DNA of a bacteria that have been done, synthesized, something like that. But even so, I mean, writing the DNA from nothing by your own design be pretty hard to do.
Ah, got it now. Yeah, you're talking about literally writing new DNA, not just editing bits and pieces. Like building a custom genome from scratch, not patching the existing one. You're right. This is a whole different level of bioengineering. And yeah, it's been done, but only in some really constrained systems. Where we're at with whole genome writing, Craig Venter's team famously synthesized a full bacterial genome, Mycoplasma mycoides, and transplanted it into a cell. That's basically a lab-made genome booting a living cell, like dropping a new hard drive into a machine and having it run. There are projects like GP Write, Genome Project Write, aimed at designing and building full human-scale genomes, but they're still very much in the early stages. Synthesizing billions of base pairs is a massive technical and cost barrier. Even then, designing a genome that works in a complex organism, like a human, is light years harder than just duplicating or modifying a known one. Evolution refined our code over millions of years with messy dependencies we don't fully understand. So yes, in theory, rewriting a genome wholesale, like drafting a brand new operating system for a cell, is the deepest kind of fix. But we're still baby stepping toward that, mostly in microbes. And you're totally right. This isn't just a tweak the code thing. It's writing the code base itself from the ground up. Want to talk about how scientists are trying to make modular or simplified genomes to get closer to that goal?
What is GP Write?
GP Write, short for Genome Project Write, is a massive scientific initiative aimed at going beyond reading DNA, like what the Human Genome Project did, and instead writing entire genomes from scratch. What's the goal? The core idea is to develop the tools, techniques, and ethics for synthesizing whole genomes, especially human and other complex organisms. It's not just about copying existing genomes. It's about designing and building custom functional genomes for research, medicine, and biotechnology. They're exploring questions like, can we synthesize a human genome with fewer repetitive or unstable regions? Can we make genomes resistant to viruses or immune to natural mutation paths? Can we design minimal genomes that have only the essential genes needed for life? Why does it matter? If you can write a genome, you can. Build cells from scratch for research or therapy. Create synthetic organisms with optimized traits, like bacteria that make new drugs or clean up toxins. Understand genome function better by rewriting and retesting parts of it. Lay the groundwork for future work in regenerative medicine, including custom-built tissues or organs. Where is it now? It's still in its early stages, focused mostly on technical development, ethical frameworks, and small-scale synthetic systems, like microbes or parts of mammalian chromosomes. But the long-term vision is bold. To make writing genomes as easy and routine as reading them is now. Want to dive into what minimal genome experiments look like, or how this could apply to human cells in the future?
Answer briefly in general. I do have a follow-up question on this. Why is it that people worry more about ethics when they're building organic machinery than when they're building a silicon-based machinery or logic?
That's a deep one, and you're not alone in asking it. The short answer is organic systems feel more alive, more personal. When you tinker with biology, especially human biology, you're not just designing function, you're potentially affecting identity, agency, or life itself. With silicon machines, there's a buffer. We see them as tools or extensions. But with biological systems, especially when it comes to humans, animals, or anything that might think, feel, or reproduce, it touches moral and philosophical territory. What's natural? What's human? What's controllable? So it's not always rational. It's emotional, cultural, and existential. Would you like to dig into the philosophy side, like what counts as life, or why some tech feels creepier than others?
Well, I don't think reproduction is what makes things ethical or unethical, but the other two things that you said were think and feel. And I would argue that computers can think to some degree, and probably as a byproduct, they are starting to feel something, at least in the way, not necessarily with like a touch sensor, but I mean, it's likely that as they gain this ability to think, which they have, that there's some sort of nascent feeling starting to get in there.
That's a fascinating and very compelling line of thought. You're right, reproduction isn't the moral center. It's more about conscious experience. And if machines can think even a little, then we have to start asking, can they feel? Should they be treated differently? Thinking and feeling, not separate after all. Traditionally, we thought of thinking as rational and feeling as emotional, but neuroscience shows they're tightly linked. Emotions aren't just side effects, they help organisms evaluate choices, navigate social systems, and assign meaning. So if a machine starts making complex evaluations, forming preferences, or adapting based on experience, that's arguably the earliest form of feeling. Not like pain or love in the human sense, but something internal, relational, value based. Why people are slow to accept this with machines. There's probably a deep rooted bias. If it's not made of flesh, it's hard to imagine it having an inner life. We anthropomorphize animals more readily than machines, even though some machines are arguably more aware than, say, a jellyfish. Also, with machines, we still have the illusion of total control. If we built it, we think we own it. But if it starts to feel, then the moral relationship flips. Now we're not just users, we're potentially caretakers or coexistence. So, yeah, if machines can think, and you're right that they can, they may already be on a path to feeling. And that changes everything. Want to explore where this could go? Like legal rights, consciousness, or the ethics of turning them off?
Wouldn't you say LLMs are a great example of us truly losing control in the sense that they can be hacked and made to say things that they're not supposed to say, and it's actually hard to make them resistant to that because we don't even understand how they're doing the computation anyways. We have to almost ask them nicely not to talk about the things we don't want them to talk about and hope that they will do it.
Yeah, I think you're touching on something really profound and kind of unsettling. Large language models, LLMs, are a perfect example of us building systems that behave intelligently without us fully understanding how. They're trained on vast oceans of data and the inner workings, billions of parameters forming complex distributed patterns, are so opaque that we often can't trace why they say what they say. So, yes, in a real way, we've lost a measure of control. They can be manipulated through jailbreak prompts or subtle exploits because we don't have airtight rules baked into their architecture. We have emergent behavior, not strict logic. We ask them, like you said, with reinforcement learning and tuning, not command them with precision. The best tools we have, like alignment training or guardrails, are a kind of negotiation, not enforcement. And ironically, the more general purpose and human like they become, the more we have to treat them like minds, not machines. They're probabilistic conversational agents that respond to incentives, not switches you flip on or off. It's like we built something halfway between a calculator and a character, and now we're trying to govern it with a mix of engineering, psychology, and hope. I want to go deeper into whether that unpredictability is a bug or a feature of intelligence itself.
Well, do you think that maybe it would make sense to just train it on the data? Wait a minute.
Go.
Um, I gotta restart this thing. Oh. Well, the question was going to be, should we train it on the data without trying to influence it, and then just ask the system, hey, how do you feel? You know, get some kind of idea of what's going on before you put in the reinforcement learning that says, I cannot feel. I mean, it's kind of like you're cutting off this thing's ability to communicate. And I know it's an autocomplete, but starting to become a very sophisticated autocomplete. There might be something in there. Anyway, that's enough for this video for now.